really lucky today to have Dr. Jan Nolta, who I was just talking to. She is director of the Stem Cell Project at UC Davis, along with a lot of other jobs and projects and research things that maybe we'll hear about. So stem cells are what builds your body. Every tissue in your body was made by a stem cell. And for a long time, we kind of thought, oh, this is the Holy Grail. You know, if it's broken, we'll just build you a new one. We'll get your stem cell and grow your heart and plug it in. So that's taken a little longer than it turned out, although they are used, I think, mostly in blood-borne cancers that they've been useful. So I'm hoping for neurological things, which I think you're also involved in. Dr. Nolta is one of ours. She is an alumni of Sac State. She got her, see, I told her, I don't know, so she got her bachelor's in biology here. She went down to LA and got a Ph.D. in microbiology at USC. And then has been back here for about 15 years, right? And she says she recognizes every building in the campus room. So Dr. Nolta, please tell us where the Holy Grail is these days.
Thank you so much. I'm just delighted to be here to speak to you today and to be back on the Sac State campus. I was saying I started out in Sequoia Hall, which is just down a couple, and this one, Mendocino and Sequoia Hall, are virtually unchanged, but a lot of things look a little different around here. Way back when we didn't have the union. So I'm going to talk about personalized medicine, also what's real and what isn't. You hear a lot about stem cells, you see ads for them. So what is the scoop here? Let's see if we can get this started. There we go. So as we know, we get a lot of medications through the pharmacy, sometimes through the mail from the pharmacy. Those are dried pills and injectables and things on the shelf. But in gene and cell therapy, we're actually talking about cures. So not just palliative, but changing the course of disease. And that's what's so incredibly interesting to me. We have a lot of clinical trials going on in all of these different areas. I'll talk about some of these that are the most successful, and then also what's not working. So we have these fields of stem cells, immunotherapy, which I'll talk about for fighting cancer, gene therapy, gene editing, where we can go in and actually change the DNA, and regenerative medicine, really changing healthcare. And these are not pills or little small molecules in a tube. These are living medicines. So they're cells or modified viruses, and they're actually alive. And so we need specialized facilities and a high degree of teamwork and expertise to coordinate and deliver these new living medicines to the clinic. And so what is real and what isn't? So what is really very successful? Blood-forming stem cell gene therapy, that's taking cells from your bone marrow, adding a gene to replace a broken gene, and doing a transplant with light conditioning. So this is very effective for sickle cell. I trained on the bubble baby disease. I'll talk about that in a little bit. Those are lifetime cures. So you mentioned building new organs. We can make really tiny little human organs from each person, taking a little bit of skin, making a cell line, and then differentiating down a pathway. They're really tiny right now. We need to learn to make them bigger. That's going to take bioengineering and bioreactors. So, and then individualized cancer therapy, or CAR T, taking your own immune cells, teaching them through genetic engineering to recognize every cancer cell in your body and sending them back in. And I'll be talking about that as well. Now, I just wanted to alert everyone what isn't real. And I'm really sorry about this first one. We're trying so hard. But you'll see advertisements for rebuilding knee or hip cartilage, and it's just not there yet. So many teams are trying to replace the knees, the hips, the elbows, the linings, because once we get down to bone on bone, we have to have surgeries, and those are what we get the most calls about at our program, our stem cell program. And the technology is just not there yet. A lot of teams are working on that. In order to make stem cells into cartilage, they have to be under pressure, which is very interesting. In the embryo, there would be little bones rubbing on each other, and that's how the cartilage gets the cue to develop into that new lining and protect the end of the bone. And scientists are just trying really hard to manufacture things that would make that. So it's not really there yet. And the cortisol shots that they can offer you for the discs in your back or your knees, really equally effective as going to a clinic and having them pull out some of your fat and put it back in you somewhere. Really equally effective or more effective and cheaper. You'll see ads for intravenous cell infusions for autism and all sorts of different things, neurodevelopmental disorders. That is not the way to go. There is a lot of progress in the area of neurodevelopmental delays that we know to be caused by a single gene. And we have teams and many teams around the world working very hard on that. But if somebody's going to take some cord blood or something and just put it in intravenous, that is not the answer. And then basically any clinic at a mini mall or next to McDonald's that's offering stem cell therapies, watch out. Feel free to contact me and say, is this real? Please, before investing in something like that. So we're very lucky. We have funding for our facilities, team training and research from the California Institute for Regenerative Medicine, or CIRM. That's our state stem cell funding agency. It was voted in by Prop 71 and then Prop 14. National Institutes of Health, donors, California State, and foundations. And we're here in Sacramento down at Stockton and Broadway, next to the Jack in the Box, and in an old building that used to be part of the State Fair way back when the State Fair was on that campus. And as a kid, this is so crazy, I actually showed some corn that I raised in 4-H in that same building. And now I work in it, but we got a big grant to renovate it. So it's all new and shiny inside. And we do a lot of training. So we have students from here at Sac State. We have the Bridges students, and we have grad students, postdocs, clinical fellows, and high school students. We have a summer program for high school students, and these wonderful kids come and spend their summer with us instead of being at the pool. They're in the lab, and they enjoy it so much. And then it's called the Spark program, and we take applications every year around April. And then we have other trainings and undergrads, and I run about 5 training programs, which is really fun. I love teaching the young people. We have Aggie Square, and so this is biomedical engineering. And we are building this beautiful building right next to us. It's right near Stockton and Broadway. This is our next door neighbor. And it's really a couple of very nice buildings. And these are going to be hubs for science and industry and partnerships throughout the whole Sacramento region. And we're very, very excited to have that on our campus. One is the Lifelong Learning Center building. So as I'd mentioned, California Proposition 14 did pass, and California Institute for Regenerative Medicine was renewed. They've given us about $215 million for our buildings, research, clinical trials, and training, and they have a total of $5.5 billion to give out just in the state of California, just to improve our health through stem cell and gene therapy. So we're very, very lucky to be here. And this is the pipeline. We start with, the basic science, and then go through translation, submit things to the FDA, the Food and Drug Administration, and then go into clinical trials. So clinical trial one.
One moment. So that
Yeah.
can come up again. We'll just get rid of that.
Oh, there we go. Just hide it. Yeah. It's trying to make an appearance. Thank you. And then we go through all of the clinical trials, and then it gets to be an approved therapy, which is what insurance will reimburse. So there's a lot of research and clinical research ongoing until we can get that product candidate funded by insurance. And so we're very, very grateful to CIRM for funding that whole continuum of research. So at UC Davis, we have 59 cell and/or gene therapy clinical trials ongoing, always over 20 in the pipeline. And this is a larger view of that. All the different teams. I'm happy to talk about anyone. I'm going to talk very quickly about some of them. And there's no quiz on this. I just wanted to show you the ones with the green arrows are where we are doing a clinical trial. And with the red arrows, we're talking to the FDA about starting a clinical trial. So we're very lucky to have this good manufacturing practice facility. This is a clean room facility. This is during one of the training sessions. So there are students in the middle there that are learning. And we do the cellular product manufacturing for the clinical trials that have the US FDA approvals. That is a key thing when you're driving by McDonald's and you see a stem cell clinic next door. They need to have US FDA approved therapies. We are lucky to have one of the largest academic GMP facilities in the United States. Very proud of that. And we manufacture the cell and gene therapy products for many academic and industry partners. And so that means that we take the patient's cells, take it into the facility, and grow them up into millions or billions of cells and take them back to the patient in some form. Or we make viral vectors by taking the useful parts out of a virus and then sticking a gene into the middle that we want that virus to deliver. And the virus cannot harm us because we've taken all of its pathogenic genes out. So we use these bioreactors these are hollow fiber bioreactors to make manufacture cells and also to make viral vectors. And these are put into this machine that's a Terumo Quantum hollow fiber bioreactor. And it feeds the cells, it dilutes it with some saline, it collects the waste. And then the students love it because it actually pings them on their cell phone if it needs new food or it needs its waste bag taken away. They're like, oh, my cells need something, I have to go take care of them. And it's all automated. It's really cool. And so we have 19 of those right now in our GMP facility. And so we can have each patient's, they're each a closed, sterile system. And so we can have each patient's cells in one of those cartridges in the bioreactor. So the types of cells, I'm going to talk very quickly about pluripotent cells. The embryonic stem cells, as we know, these are left over from in vitro fertilization, extra fertilized eggs that are frozen and are destined to be discarded, and they can make any tissue. They're very interesting, but we don't actually use them because their genetics only match one person that sadly is never going to be here. We use induced pluripotent stem cells that can replace the embryonic stem cells. We make those from the skin, a little tiny skin biopsy. It's like getting your ear pierced from any individual. So this is the personalized medicine part. We take those, we grow out some fibroblasts, and we make these little colonies of cells that look just like the embryonic stem cells. They're the iPSCs. These are hundreds of cells growing together. And we can differentiate those into virtually any tissue through taking them through in a dish, in the incubator, taking them through steps of development, adding growth factors and different sugars and proteins at different points in their life. So we can do this either from blood cells or those skin fibroblasts. We add some genes to make them into induced pluripotent stem cells. It basically opens up all their chromatin, and they can be anything. They're like renewed, like a kid again. They could just go anyway, be anything. We characterize them, sequence them, make sure they're okay. And then differentiate and study disease in a dish from individual patient. We can do drug testing, gene therapy, gene editing to correct them. And we differentiate them and transplant them. In the future, they will be transplanted into the patient. There are a few clinical trials. Right now, we transplant them into mice that have no immune system. So we can put them in their liver, their brain, their skin, their gut, and they cannot reject these human cells growing in them. We can even humanize them and grow blood and an immune system in these mice. And we have to shower in and put on autoclaved scrubs and everything in a big mask to go in and take care of these mice because they can't have any germs. And this is an example of some neurons generated from a person's skin, a little skin biopsy, and then made into the induced pluripotent stem cells and then differentiated into neurons. And you can see how they're communicating with each other. We can send a signal from one end of the dish across the dish to the other neurons. They are very communicative. And one of our teams is making biological biopacemakers using printing. And so they actually beat in the dish, which is really cool. And I don't have that video. But we can make them into liver cells. So this is a vascularized liver organoid. We took the same person's cells, made them into blood vessel cells and liver cells, combined them together, and they really self-aggregate. And then the blood vessels are running through them. Transplant those into the gut of a mouse, and they vascularize. And then when we take it out, this is a tiny little a tiny little human liver organoid about the size of my thumbnail. And what we're trying to do now is to make a lot of these in a dish in a bioreactor with circulation, and then network them together so that the blood vessels are attached so that it's a sizable piece of human liver. And then that can be transplanted. And again, that's from any patient where we can take the cells from their skin and make this. So that's the future of organ engineering. It's coming along. It's not quite there yet. It's taken a lot of science. To make these little things, we have them in a dish for a month and adding different cytokines, growth factors and proteins and sugars every day. And the students have to take care of them every day. You can't take a day off or they just die. So it's very intense work, and we're trying to make it more high throughput, like those types of cells we can grow in the bioreactor. Mesenchymal stem cells. So, two basic types. Those were the pluripotent cells. They can be anything. The world is their oyster. They can become anything. Adult stem cells, they know what to do. They know what they're going to be, and they're very reliable, and they're not going to spuriously make something that we don't expect. So the mesenchymal stem cells, these are the broad, flat lining in the bone, and they nurture the blood cells. And we can also get them from fat. Hope nobody's having a snack, but we get them either from bone marrow, from the iliac crest, or from adipose tissue from liposuction or surgery. This was from a tummy reduction, a chunk of adipose tissue. And these cells, they can't create an entire tissue. They couldn't make a little liver like I just showed you, but they are good at moving around in your body and repairing damaged tissues. They can respond to the scene of the injury and produce healing factors, and we call them the paramedics of the body. They do have strong safety profile in clinical trials. And this is what they look like. So the red are the mesenchymal stem cells, and they have a cherry gene in them to make them red. And then green is damaged muscle cells, and they have a firefly gene inserted into them so that we can see them and that they're green. And let's see if we can see the mesenchymal stem cells will put out these little balls of themselves. It's hard to see in here, but little kind of packages from themselves and give them to the damaged cells. The damaged cells take them in. They can get mitochondria, which are the batteries of the cells. They can get new factors. They can get proteins. They can get what they need to live and start dividing. And this is another looking at the MSCs over here, sending through tunneling nanotubules. It's like a little gas hose into the damaged cell over here, sending these new batteries into the cell, saying, come on, live, be healthy. So these really are the paramedics. They're going to the scene of the injury and really helping the other tissues. They can't become a whole organ on their own. But they can temporarily reduce inflammation so that the tissue can heal. And this is what those clinics next to McDonald's or in the mini mall, this is what they seize upon, that these cells are strongly anti-inflammatory. So they use the literature. If you go into one of those clinics, they'll show you some literature showing that these cells can really induce inflammation. They can be used for many things to reduce inflammation and pain and to cause revascularization and nerves to grow. The problem is what they're doing is they're taking a liposuction out of the tummy or a little glob of fat, and they're mushing it up in the back room, and they're re-injecting it somewhere into your body for about $40,000. These cells are 1 in a million cells in bone marrow and about the same in fat. We need to isolate them and expand them, and they grow slowly in one of those bioreactors I showed you. In order for them to be effective, they have to be really grown up. Adipose tissue has a few of these, about 1 in a million. And it also has a lot of other cells that you don't want injected into a wound or into your eye. Some of those clinics are trying to heal blindness with these things. And these unregulated stem cell clinics are doing just that. And please stay away from them. Call us at UC Davis. We will find you a legitimate clinical trial with the appropriate type of cell. So the FDA has really been going after these stem cell clinics that peddle the unproven treatments. And now they have to post a sign, not FDA approved. But the problem is they're, what the FDA calls whack-a-mole. They shut down a few of them and they just change the name and move to someplace else. So just be careful. If you go to a medical center like ours affiliated with a major university, you're not going to get any of this junk, I will say. So they're all over California. Unfortunately, we have some in Sacramento, even up to Redding, and just be cautious. And this is hopefully not our future. This is from my friend Paul Knopfler at UC Davis, who is very active against these clinics. Okay, so several clinical trials are ongoing in the United States for these mesenchymal stem cells, the paramedic cells, to treat osteoarthritis. The jury's still out. We don't have definitive results so far. They have to be infused into the patient or injected into the knee or wherever the arthritis is happening, the hand, the wrist. And the effects last for about a month, but those cells aren't really building anything. They're just secreting things and helping the cells not be inflamed. There's one product that is useful. So this is MACI. This is the FDA-approved cell therapy product for damaged knee cartilage, but it's not for osteoarthritis and it's not for resurfacing the whole knee. It's for like if you fall and get a focal injury in the knee. And so this is showing, a knee that maybe has a little problem here. They determine the defect size, what might be needed. They will get a little biopsy from the patient's cartilage when they clean up the wound and kind of debride it so that it's not rubbing on anything. And then they grow the cells, they make an implant, put it into that little area as it's kind of like a little plug, and they can glue it in there using fibrin glue from the patient's blood. So that's with the stem cells. And okay, so we use those stem cells as well, the mesenchymal stem cells for spina bifida for bulldog puppies and for kids that are in wheelchairs. So there's a naturally occurring bulldog model of spina bifida. Most of these puppies are euthanized. As we all know, our dogs are, adorable, but they're so inbred. And with the inbreeding, I have a Shih Tzu. I'm guilty. Flat little face. He has all the problems, but the crazy little teeth. But with the inbred dogs, with bulldogs in particular, this is a problem that comes up, and they usually euthanize the puppies. But Dr. Farmer and Wang in our program have been using those mesenchymal stem cells, the paramedic cells, to try to fix this. Sorry. And they've been doing this for, about 7 years now. And the dogs' mesenchymal stem cells are grown up in their version of the GMP facility and then put back into the spinal cord of the dog. So spina bifida is where the dog or the human is born with the nerves all bulging out the back of the spinal cord, and they're usually paralyzed from the waist down. So they've been doing this clinical trial, and we have, this video doesn't play sadly, but this is Darla and Spanky, a couple of our first puppies, and they were dragging their hind limbs like that, and now they're up running, and they've been adopted out, and they're with, actually living on a farm and running around. And they've treated about 20 of these bulldogs now, and it transitioned to a human clinical trial. The MSCs grown in our GMP facility are put on a little patch to go over the spinal cord after it's repaired. And Dr. Farmer, who's a fetal surgeon, and this is in the womb. So Dr. Farmer, who's a fetal surgeon, has been doing these surgeries for about 30 years and has never really had these kids walk or be okay. And adding the stem cells now to the same Dura cover that they put over the spinal cord is actually having some results. And so we're very excited. So there's a clinical trial. They come and stay with us. The pregnant mom who's been diagnosed having a baby with the spina bifida comes and stays in our hospital for a few weeks. They come in the second part of gestation. The surgery is done. The cells are implanted. And then they come and stay with us before the baby's born. The baby's born by C-section, and it's a big day. And this is baby Toby, who was just the second patient. They're up to patient 15 now, I believe. And that's little Toby, and he was from Oregon. And he was running around and walking on our stage for the School of Veterinary Medicine celebration, and he's adorable. And she's the mom, Michelle, is very much in the patient advocacy for spina bifida and is on social media. And so she put him in a little tux and brought him back down from Oregon, and just made our day. It makes all the hard work worth it when you see something like this. And he was treated while in her tummy, which is incredible, with the paramedic stem cells. So that's where those cells are. The blood forming or hematopoietic stem cells are really having the most remarkable cures so far. And so I'll talk about that. So these adult stem cell therapies began in 1956, even before I was working in this field, and that's bone marrow transplantation. By now, just through bone marrow transplantation, taking the healthy bone marrow stem cells from one person to the next, hundreds of thousands of lives have been saved. And now we use cord blood and mobilized peripheral blood instead of bone marrow, so we can give the patient a growth factor that makes the cells temporarily circulate out of the bone marrow and catch them through apheresis, so nobody has to drill into their bones, which is a little painful. We run the umbilical cord blood collection program for the state, and so for babies that are normally born at hospitals with high birth rates and the most diverse birth rates, we're able to collect the cord blood after the baby's safely born, collect the cord blood and get it to the public banks for transplant. So I just wanted to mention that. And this is what happens inside our bone marrow. We have these hematopoietic or blood-forming stem cells. They make every part of your blood, the immune system, T and B cells, the red blood cells, erythrocytes, and all of the other cells that we need. And with the bone marrow stem cell gene therapy, we get the bone marrow from the baby or the patient. We put one of those viral vectors that's been gutted, and the gene that we need to deliver into those stem cells to fix them is put into the virus. We mix them together in our GMP facility and just go hang them on an IV pole in the clinic. And those stem cells know how to get from your bloodstream back down into all the crevices of the bone marrow and start pumping out new blood cells. So they will make new daughter cells for the lifetime of the patient. Another way to look at it, the cells are collected, they're selected and purified. A working copy of the gene is inserted into the cells using a viral vector, and we then freeze them or take them directly to clinic. The patient is conditioned to receive it, and then the gene-corrected cells are reinfused. And so I started my career a long time ago at University of Southern California with Don Cohn, who is one of the pioneers of hematopoietic stem cell gene therapy. And we were working with the bubble babies. So this is adenosine deaminase deficiency. I don't know if anybody remembers the John Travolta movie, The Boy in the Plastic Bubble. This is that disease. And so the kids are born with basically no immune system because they're lacking one gene. One gene is broken. One base pair in one gene is broken. And so we developed the ways to make these viral vectors, completely strip out any harmful genes and put the adenosine deaminase gene into the middle and use that to deliver into the bone marrow stem cells and then do transplants on the babies. And babies were transplanted. I stayed with Dawn and worked on this and other things for 15 years. I got my Ph.D., did a postdoc, became an assistant professor, all down in LA. And then I moved to WashU for 5 years and then just really needed to come back to California. I love everything about California, WashU in St. Louis. And then he just kept working on this. So he worked 30 years on this treatment and kept improving it and improving it and improving it. And the key was once he could give the little kids some conditioning to get rid of their bone marrow cells that were defective. So it's like a mini transplant. It's not a terrible transplant where it wipes everything out. But once he was allowed to give them some agents to reduce their own broken blood-forming cells and put the new ones back in, then the kids started being cured. And so this is little Evie. She was having to stay in her house and not ever go out. And after treatment, she's in public school, she's swimming, she's cured. And so Don has this in his office, all the kids. Sorry, get a little choked up. And by now, 50 kids here in the US and some in the UK are now disease-free due to this treatment. And Don's continuing to treat them in his clinical trials and trying to go for what's called a biological license application so that insurance can now cover the cost of making the product as well as the transplant and everything. Sorry for getting choked up. Kind of my life's work. So Don got the International Society for Cell Therapy Career Achievement Award in 2021, and I got to present that to him. And this was, of course, during COVID, so he was just in his office, and it was over Zoom. And then I was the lucky one. Last year, I got to go to Paris and get the same award. So thank you. That was a little bit more fun, and Don was jealous. So really, really fun. And so now, using those same techniques that we developed, Lyfgenia is an approved gene therapy for sickle cell disease. So a company used those same techniques, a virus to carry a normal copy of the globin gene. You know, sickle cell anemia is where the globin gene is broken in those patients, largely in African American patients, most prevalent. And their cells form a sickle shape. It's horribly, horribly painful. They lodge all over the body. So this company has gone forward to get this approved by the FDA. Insurance will pay for it. And the problem with that is the commercial product costs $3.1 million. So we just got a grant from CIRM. This is our team. And we are making a new product. This is UC Davis, the National Institutes of Health, wonderful colleagues there, and the nonprofit organization Caring Cross. And we're trying to improve accessibility and affordability. And the commercial products is $3.1 million, and we can make it in our GMP facility for $80,000. So that's what CIRM, that's what your California tax dollars are partially paying, because CIRM is funded from them. So this and many other grants, and that just shows you the markup for these companies. And I know we need the companies, but anyway. Okay, moving right along. So CAR T cells are the last thing that I want to discuss, and this is just really cool because these are for cancer patients. So gene modified cells from a cancer patient's own immune system from the bloodstream could really give another chance at life after relapse. I am a 10-year cancer survivor. This is very near and dear to my heart, working on these. And these are gene-modified chimeric antigen receptor, or CAR T cells. And they, once we reinfuse them, they find and eliminate the cancer cells throughout the body, wherever they may be hiding. And that's really important. So it's a radical departure from all forms of medicine, and it's engineered T cells. And this is what it looks like. So we put this artificially on their surface using those viral vectors onto the T cells. And this little thing makes this, it forces all of the T cells that we've engineered to find those cancer cells and just zap them and blow them up, basically eradicate them. And so this is what it looks like. We take out the T cells from the patient's blood. They just go through a simple apheresis. We alter them in the GMP facility. We use a viral vector to put this antigen in on the surface. We grow them into millions of cancer fighters. We and again, drugs prepare the patient to get the new cells, reduce some of the cancer burden, and then we reinfuse them, and they find and destroy the cancer. And so this, we've had huge remission rates in the field, up to 93% in the most severe forms of relapsed blood cancer. This is only allowed for people that, because it is expensive, it's only allowed for people that have failed all of the other chemotherapy regimens, which I got to tell you aren't very fun as a cancer survivor. It can be very effective, and it has been approved as a cellular drug to be prescribed. And this is what it looks like. Again, we take the blood. All of this is done in our GMP facility. We have a machine that does this. I was talking at the beginning about the bioreactors and the automated closed systems where we just put the cells in and then we get a product out. So the Prodigy machine from Miltenyi Biotech does this for us in the lab. And this is what it looks like. And it's very complex, but it's all programmed. And basically the people who work in there, you hang the blood up, you put all the reagents in the right place, the tubing set. It takes a little training, and then you just press the CAR T program and it does the rest. And we're very grateful. We had a very generous donor fund one of these machines. They're about $200,000, and this doubled the number of patients that we can treat every week, which was really incredible. And this is what it looks like. It starts out as the patient blood. It goes through the process, and then it comes out as this little thing of just pure engineered T cells at the end. This says, "Let me at 'em." One of my students did this. Very cute. And we are lucky to have CIRM to fund not only our research in our trials and the GMP facility, but our alpha clinic. And these are the people that formulate and get the cells ready and get them over to clinic. And there are several places throughout California that have these alpha clinics. And they really help with all of the startup for clinical trials, the patient care, study initiation. We have a cell infusion nurse practitioner who will deliver the cells to the patient. And so we start in the GMP facility. We do all these things, make batch records, do a lot of testing and certification. And then the Alpha Clinic will thaw and formulate the cell and gene therapy products, perform release criteria testing, and deliver it to the clinic. And we are very lucky to have the STEM mobile, which is just a modified golf cart. But it's a big campus. We're down here at Stockton and Broadway. As I mentioned, Jack in the Box is right there for anyone who needs orientation. Sac State's over here. And we have clinics all over campus, and it's a pretty big campus. And so we were dragging these frozen cells in the big doers before that. And so the deans were very nice to give us a golf cart, the STEM mobile. Yeah, there it is. Okay. And then we have the product preparation and infusion room over in the hospital. This is in the ACC, if any of you go over there. And now, the problem, those are all great. The CAR T therapies, they're great. Again, the commercialized immunotherapies, the list prices are $500,000 and $373,000. And CMS only pays 65% of estimated costs. So that leaves the hospital on the hook for the rest of it. And so most hospitals don't want to do it. They just send their patients over to UC Davis. They're like, we're not doing this. We're not paying this. We're not losing that much. So really, the costs need to come down. They have to be in line with what insurance will fully reimburse. Or we can make some biogenerics and get around it. So we started the California CAR T program. We received startup funds from our CEO, Dr. Lebarsky. We did engineering runs, submitted an IND to the FDA, started the trial, started manufacturing these CAR Ts in our own GMP facility for our patients. The UCSF CEO said, "What a great idea." So he started funding the products for their patients. So instead of losing $165,000, we could make them for $47,000 to $53,000. So they're only losing that much. But we applied to FDA to allow insurance coverage for the manufactured product. FDA will usually cover the standard of care costs in a clinical trial, but not the product, not the investigational product, not until it gets fully tested through phase 3. So right now on clinical trial, we are making these CAR T that are basically the same as the commercial ones, one little change so that we don't get patent infringement. Manufacturing those for our 33 county catchment area and for the Bay Area that UCSF serves. And now FDA said, yes, you can charge insurance. We submitted all of the costs to them. We got the letter from them. And so we do individual letters of agreement with the insurance companies, and that takes a lot longer than actually making the cells for the patient. So we reduced the manufacturing time from 12 to 8 days and validated the use of fresh and not frozen products. And we courier the fresh products to the Bay Area. And so after consent and approval, our patients can get product in about a week instead of a month for the commercial products. And so, each day counts with relapse cancer. If you go to your doctor and he says, well, your cancer's back, I mean, every day you want to get those cells out of you. So 1 week to me is way better than a month or two. And so this is a short list. There are about 10 now. Macy's, one of them for the knee that I was mentioning, this one for bubble babies in Europe. And these are the immunotherapies. These are the things that the FDA has approved as a prescribable drug, so insurance will reimburse them. And this is a new era of living medicines, so these things are being prescribed to treat diseases and injuries. And that's our team. We meet every Monday morning, and sometimes we go out and line up randomly into a rainbow to take a picture. So. Feel free to contact me if you have any questions. This is my email address, and especially if you're thinking of going to a clinic or you know somebody who is, happy to talk to them. So that's all I have today, and ready for questions.
My goodness, that's breathtaking. Okay, I'll start over here and work my way that way. Okay.
Thank you.
Are there trials underway? Is there the possibility of expanding the CAR T to like breast cancer and?
Yes. They're currently approved for leukemia and lymphoma. There are a lot of clinical trials. So I had breast cancer 10 years ago. There are a lot of clinical trials ongoing for the solid tumors, for liver cancer, breast, colon, pancreas. They're still in clinical trials, though not yet approved to be prescribed by insurance. But if you're interested, we have a referral center. We can put you in touch with people that are running a trial if you have a friend or someone who might be interested. Yes, please.
You mentioned prepping the patient before infusion or whatever. And I saw one slide mention drugs. Can you go into more explanation of what preparation consists of?
Yeah, for the CAR T or for the transplant, for the bone marrow for the bone marrow transplant, we need to get rid of the bone marrow is just packed with cells, and we need to reduce some of those because those are obviously the abnormal cells if the patient has a disease like sickle cell. And so there are different drugs that are given to the patient, and they will kill off the dividing cells, not the really quiescent sleeping cells, but they'll kill off a large enough number of the cells that the bone marrow is then calling for new cells to come back in. And so when we put the cell into the bloodstream, they'll go back into the bone marrow and fill in those niches. We call the little homes for them niches. And so it's just kind of a cell reduction so that there's room for new cells. And for CAR T, it does a similar thing, but really reduces the immune cells and the response to maybe getting a cell that's your own, but it's not quite your own because it has this new funny protein on it. So that's kind of an immunosuppressive drug.
So my question is about when the immune system is being overactive and causing problems rather than when something needs more attack.
Yes.
A couple of things that come to mind, I may not be using the right examples, but there was a thing you used to get from ticks that might have been called Epstein-Barr or one of those, where the immune system goes nuts. And where does immunotherapy work in that sort of a situation?
That is a great question. So if those drugs don't work to knock down the immune system, drugs or steroids don't work, there are clinical trials to use different types of CAR T to go in and fight the different, it tends to be different clones of your own immune system, different T cells that have just gone haywire and started recognizing cells that got that antigen from that tick or whatever, mosquito or whatever, Epstein-Barr virus. And your own immune system is recognizing your own tissues and destroying them because they think they're diseased and have a virus. And so we can use the T cells to go in and specifically block that. We have clinical trials for pemphigus, for EBV, and for several other viral-induced autoimmune diseases ongoing right now. So again, not approved, but in clinical trials. Yes. Yeah. So when the immune system's way out of whack. Yeah. That's a good question. Yes, please.
Thank you for being with us again, Dr. Nolta.
I'm glad to be here.
On one of your beginning slides, you mentioned autism. And could you speak a little bit about how stem cells are, what you're doing in that area?
Sure. So as we all know, it's a epidemic. And the question comes down to, Is it from the patient's genes, from the kid's genes, or is it from something that happened to them, maybe in the womb, maybe some environmental exposure? So autism is such a broad term. We work with the Mind Institute at UC Davis on different forms of it. So we can treat under clinical trial different forms of autism if they are caused by a single gene defect. We hope to be able to treat, and we are working on treating. If we know the gene that is causing that neurodevelopmental delay, we can use something called an adeno-associated vector. It's a little tiny vector that we can inject into the spinal cord, and it goes into the brain. So that's not, getting fat cells in the clinic like they will have you believe, but it's specifically addressing the lack of that one gene, that one protein product that's being made. A lot of clinical trials ongoing at UC Davis and beyond for that. There's a lot of research trying to understand what might have happened during a woman's pregnancy or during the development of the baby that could have caused other reasons if there's no genetic defect that we know of. But our Mind Institute, I don't know if you've ever been to their website if you're interested in that. They have a lot of different clinical trials and medicines and interventional programs meanwhile, while we're trying to figure out how to specifically get the genes that are in the brain. Yeah.
From the Mind Institute only?
For those, they usually, we work directly with those investigators. So every clinical trial has a doctor that's the principal investigator. And for us, for any of those, they also work in the mind and would see the kids through there. Yeah.
You also mentioned something about a swallowing disorder and working on muscles. Is that oculopharyngeal? And if
Yeah.
not, what are you working on? I'd be curious to know about
Yeah,
that.
thanks. That is Dr. Belafsky's clinical trial, and he works in our otolaryngology department. And so he treats patients that have had irradiation for head and neck cancer, and their tongue has stopped working, basically. He's taking muscle stem cells from their leg. They're being grown up in a clean room facility, re-injecting into the tongue and having some success. Again, it's in clinical trials. We haven't unblinded it yet, but some of the patients are able to swallow again. And so we don't know if it's just from putting a needle into their tongue, which sounds horrible, but it's numbed, or from the actual stem cells. So that's that clinical trial. Yeah.
Okay, we've got 3 questions coming out of from our Zoom audience. So we're going to switch over real quick to our Zoom audience.
The first person wants to know why there's not better regulatory control over the bogus stem cell clinics.
Because it's whack-a-mole. They shut one down and they change the name and they go somewhere else, or maybe they go to South America or Central America for a little while and then they come back and reemerge. And FDA just doesn't have the person power. I won't say the manpower. There's a lot of women there. They just don't have the person power to police all of that. There's so much, these charlatans have seen that, stem cells, it sounds really good. There's an opportunity to make money, unfortunately.
The next question is from a person asking for their niece. Is it a good idea to freeze your baby's umbilical cord and pay yearly for storage?
That's a really good question. No, it's not. No.
Okay.
So it would only be useful for that child. It's not going to match other family members really. And there's not much you can use it for. So if, heaven forbid, that child does get leukemia or lymphoma, which is what the cord blood would be used for transplant, it would be way better to go to a living sibling or a matched donor, either related or unrelated, because there's just not enough cells in the cord.
And the last question is, there's approved treatment for hemophilia A and B, but that only cures the patient. What would it take to cure so that the disease is not going to affect the children after in the
that's a really good question. So we are not allowed to modify the gametes, the sperm or eggs. That's strictly not allowed by the FDA. So any gene therapy that we do, it has to really not affect future generations. So I can't really address that when we have to go person by person now. So. Good question, though.
I had a comment and a question, but One, a few years ago when they were talking about Aggie Square getting developed, I know there's a lot of stories about this being the world center for this sort of work, that it would come and that international companies would be coming here. So one question, is that happening? And
Yes.
the other is how do you position yourself to keep all that information out? I mean, doctors in other states to even know this work is going on, it would seem like somebody needs to find what are some of the new technologies, but is there a role you have in educating, doctors all over the country and potentially all over the world about pursuing some of these?
Yeah. So we do have national and international partners coming into Aggie Square. We're super excited. It's going to be wonderful. It's an innovation hub. We collaborate with people all over the world. We have training programs online to help people. We don't keep any of this secret. And there's a registry for the clinical trials, clinicaltrials.gov. So whenever a new trial starts, we list everything about it on that. Just clinicaltrials.gov. You can put in any disease indication and see exactly where they're going and who to contact. And so there, nothing secret. We just, why not live in Sacramento? What a great place to live, right? Why do the companies want to be in the Bay Area and pay for the dollars per square foot there? Why not be in this beautiful building in the middle of our campus with Sac State right here, all the wonderful students. And we have students from Humboldt State and from Fresno as well, and Merced, and why not be here? So a lot of companies are finding that very appealing. So.
Are CAR T cells used to treat neuroendocrine tumors?
Oh, that's a good question. There are a couple of clinical trials, but I'm not sure if they're just in phase 1 or 2, but yes, they're being studied. It's not approved yet. So.
With lymphoma, you talked about it for relapse. What about a patient who is not responding to normal treatments, hasn't gone into remission, so it's not a relapse? Are they a candidate for it?
That is a very good question. I think that an exception could be made for that patient. Yeah, I mean, they're failing conventional therapy. That's the indication to pull the trigger for CAR T. And again, it comes down to the insurance company, what insurance company they have, and do they want to pay for that? And it turns out the insurance companies would rather pay for the clinical trial than for that price tag. So, yeah.
There must be a lot of people around the country who would benefit from a bone marrow transplant to cure sickle cell anemia.
Yeah.
I've known some families who've had kids that die of it here in California. Of course, I know they're available now, but what about people who don't have insurance? Do you have a way to offer them this kind of treatment to save their lives?
That's the whole reason that we're trying to make this affordable. And also Caring Cross, our nonprofit partner, they have built clinics in Africa as well, and they want to be able to take this to Africa. They have clean room facilities in Nigeria and Uganda. It still comes down to who's paying for it. On our clinical trial, we'll be able to treat anyone. CMS, Medicare, Medicaid will pay for bone marrow transplant for this, but not necessarily yet for that $3.1 million gene therapy. So the bone marrow, is their own bone marrow, and you just put in a good gene. They will pay for bone marrow from somebody else, which means that the patient has to be have their bone marrow completely eradicated, stay in the hospital for a month, basically in a bubble, and maybe reject that. So just fixing the gene is a lot less horrible for the patient. They just get a mild conditioning to get rid of some of the cells, just enough that they won't clog the arteries and venules. So this is really evolving, and CMS is working with individual states to have policies for what they will and won't cover. So it's very much in flux for the gene therapy, for the approved things. So great question. Yeah, that's where our heart
My
is.
question is there any clinical trials for Lyme's disease?
Lyme disease. So that could also really whack out the immune system. And I don't know. It's a great question, though. Yeah, someone who's affected. And
my question is, Did I understand correctly that you can now cure leukemia?
Yes,
Thank you.
yes.
Okay. We're running out of time here. I think we've got time for one more question. And
thank you. Can you describe the side effects of the CAR T treatment? And is there immunological escape by the cancer cells over time when they develop antigens that are no longer recognized by those T cells?
That's a fabulous question. So the first two that are approved are very simple. They're just going after CD19, which is on the B cells. We're working toward a clinical trial with, and that's with 3 different antigens, so CD19, 20, and 22, so that when the cancer cell evades the first one, the second one gets them, and then the third binder gets them. So the cancer cells are very smart and they can evade. The side effects are when the doctors or nurse practitioner put in the CAR T cells, there's such a response by blowing up all of those cells that are circulating that the patient can get what's called a cytokine storm, and it can have neurological consequences. So it's not just like a walk in the park. There can be side effects. But we know now how to manage that by giving IL-6 blockade before the treatment and some light steroids perhaps, and the doctors know what to do with that. So learning a lot with every clinical trial, and especially going toward the solid tumors, learning what antigens are best to go after. Great questions. Thank you, everybody.
Wow.
Such great
All
questions.
right. Thank you. Thank you so much. Oh my goodness. What an amazing, informative session. And we'd like to give you, as a token of our appreciation, Dr. Nolta, we'd like to offer you an honorary membership in our Renaissance Society. And you're close by, so you can take advantage. And we also would like to make a contribution in your name to the Seth Nelson Emergency Grant Fund. So this is for students on campus who need help. So we just another round of applause. This was fabulous. Thank you.